Case Western Reserve University's Institute for the Science of Origins proudly presents Real Science Now, featuring top experts in science and medicine, covering everything from new planets to curing cancer to virtual reality and many topics in between. The Real Science Now lectures are hosted by the Great Lakes Science Center and presented with the assistance of Case Western Reserve University, its College of Arts and Sciences, and MediaVision. Thank you for joining us. My name is Glenn Starkman, Director of the Institute for the Science of Origins. Today's program features 3 presentations entitled Brain, the link between stroke, aging, and diet, The New Childhood Illnesses, Addiction, Autism, and Lead Poisoning, and Human Evolution, Recent Advances in Unraveling the Complexity of Our Ancestry. Let's begin with Dr. Joe LaManna talking about the human brain.
Today I'm going to try to use some specific results from our own work, but also to generalize to some concepts that I think would be useful for a wide range of ideas on neurodegenerative diseases. So, no surprise to anybody, the population is aging and aging rapidly, and this is not a bad thing. This is a good thing. This shows that we medical researchers have been successful. I made a calculation. I think that the average lifespan increases by 3 months for every year since 1950, since NIH was invented. So this is a good thing. However, aging is a major risk factor for quite a few diseases, such as stroke, cardiac arrest, dementia, and other neurodegenerative diseases, and others. These are my favorite ones. And the question is why? So the prevalence of these diseases is becoming more and more, but that's because we're living longer. But why is this happening? Well, let's talk about the brain economy. I tried to figure out a relatively simple way to explain a very complex system. And here, the brain economy is based on supply and demand. That is, demand is the nerve cells. They're the ones that are having synaptic activity, doing our thinking for us. And that's the work that the brain does. Now, on the supply side, we have the vascular system delivers substrate in the form of sugar. For the brain is mostly sugar. And it's burned by oxygen. So both oxygen and sugar have to be delivered by the vascular system of the brain. So it can be used in mitochondria to generate the power that supports the work of the brain. So the supply is provided directly by a distribution network. And this distribution network is especially important part of this is the small vessels, the capillaries. So the vulnerability of the brain is basically a supply-side economics problem, and the distribution system, which are the capillaries, needs to be preserved and protected in order to maintain the brain healthy. And today we at NIH especially, we recognize that to talk about brain function, you have to talk about all the cells in the brain. The neurons, the glia, the capillary cells, and the pericytes, which are stem cells that live with the capillaries in the brain. And that these, the neurovascular unit dysfunction is important in all the diseases that you see there in a wide variety of conditions. So, by the way, this talk is about the small vessels, these capillaries. And what I'm showing you here is a plastic cast made of the vascular system of a rat brain and in different magnifications. So the one in the middle there with the white box in it is a cross-section, and that strip going in a curve, that's the cortex of the rat. And the box is then blown up on to the right. So the figure on the far right is a blow-up of the box, the white box there. And all what you see, all that system, that's all capillaries inside the brain. And the blood supply comes from the top down. The bigger vessels on the right are in the pia, the arteries. They penetrate, and then they break up into lots of capillaries, and then the blood gets collected by the veins and brought back up. And as a very important part of that, besides the delivery of the oxygen and the sugar, is the protection of the brain from the rest of the body by a barrier, the blood-brain barrier. And this is made up of astrocytes, the green star-like cell, hence astro, which surrounds the capillaries and induces the capillary cells to make a barrier. So this is a cooperative event. You need the astrocyte to give a signal to the endothelial cell to be a brain capillary and not any other organ, and to make a tight junction. Along the capillary, along with the endothelial cell, is a pericyte, and the pericyte is an undifferentiated cell that produces lots of different growth factors and participates in the signaling. So the point here is that the function of the brain involves not just a nerve cell talking to another nerve cell, but also communicating with hormones and signals and chemicals to its supply support system and the capillaries. And all of those signals need to be maintained, and we don't know them. We're just learning about them. Because we thought, this is, by the way, an EM picture. So this is one single capillary cut and cross section on the left there. The L stands for lumen. That's the center. That's right above it, that dark mass. There's the nucleus of the endothelial cell. And then the single endothelial cell. So the capillary is made up of one cell that wraps around and grabs onto itself and has the barrier. So on the right are examples of the tight junctions that hold the tissue together. And we thought for a long time that once you grew these capillaries in the adult, they were there, they stayed, they were permanent, they were a permanent structure. And in fact, what we now know is that they change in response to environmental changes, such as altitude, or demand changes, such as changes in neuronal function. And the first time we saw this was an experiment that we did in the 90s, where we put the rats into Dr. Decker's chamber. And it was his chamber at the time, working with Dr. Stroll and myself. And after 3 weeks, we found that the number of capillaries in their brain was about 3 times, was increased by almost double compared to their litter mates that had stayed at sea level. And this was amazing to see this large increase in the number of capillaries, this angiogenesis that was stimulated by the hypoxic experience. I'm not jumping way ahead. We also have found out that it's not just the hypoxia that induces the increase in capillaries, so 2 different state changes, but it's a continuum. So that is the amount of the capillary density is dependent upon the oxygen availability in a dependent manner, not 2 states, but continuous dependent manner. So these were mice in this case that were put into a high oxygen atmosphere, and in this case 50% oxygen for 3 weeks. And over the 3 weeks, their capillary density decreased. By the way, their hematocrit decreased as well, the red cell. So this is a continuous signal that is adjusting the capillary density to the proper delivery of oxygen. What else can change capillary density? So it's not just the availability of oxygen, it's the matching of what's available to what needs to be used. So if you take some mice and you put them into an enriched environment, the one on the left is the normal laboratory environment, normal cage size, all approved and appropriate, and what everyone uses in their labs at the facility, in the animal facility. The one on the right is the luxury suite version of that. It's a larger space and it has toys in it. And the toys are changed every few days or so. So this is an enriched sensory and motor environment. And the black bar on the far right there is the increase in capillary density that's induced because that type of environment gives you increased neuronal activity. Sensory and motor activity is increased on a regular basis, capillaries go up. How does this happen? Well, what we know now, we now have a good idea. Starting in the 90s, Greg Semenza at Hopkins started looking for what was the transcription factor that stimulates the increase in red blood cells when you go to altitude. So what increases erythropoietin, which is generating more red cells? And he found the hypoxia-inducible factor system. And this is how it works under normoxia. It's a little bit unusual because this is a system that continually makes a molecule, HIF. But in the presence of oxygen, that HIF is hydroxylated by the enzyme prolyl hydroxylase, and then degraded. So it's made and degraded, made and degraded. As long as everything is fine, made and degraded. As soon as the oxygen falls a little bit, it builds up. Once it builds up, goes into the nucleus and stimulates now somewhere up 100 genes or so, mostly that, oops, unbelievable. I have it on silent mode, but it's, you know how a family emergency breaks through. So, anywho. So this particular transcription factor was the one that was, it's an ancient system that switches between anaerobic and aerobic metabolism. So it's in all cell types. And what happens is, as it gets activated, it releases VEGF, vascular endothelial growth factor, which acts as a stimulus to grow more capillaries. If, however, it is activated without the growth factor, then it will cause the cell to die, because it causes a disruption that in the presence of a growth factor leads to growth, in the absence of a growth factor leads to death. And that's how you regulate the amount of capillaries that you get. Now, if you do increase the number of capillaries, you can protect against stroke. So the more capillaries the rat has, the more of a protection they have, because they have increased watershed flow. There's more vessels. So the stroke is smaller. There's a work of Michelle Pukowitz at Case. These are, show some pictures of the people who did the work while in the lab or collaborating with me. And this was where these rats had been on a ketogenic diet, which induces the capillaries to grow and HIF to go up. HIF goes up, capillaries stay perfused, and it protects against stroke. And in cardiac arrest and resuscitation, work of Kui Shu in the physiology department, where the rats on the ketogenic diet lived longer post-stroke, post-cardiac arrest and resuscitation. Again, with HIF going up. Juan Carlos Chavez noticed about 15 years ago that the older rats didn't produce HIF. Don't worry about that's the data. You could look at it later if you need to. But what it shows is that if you chemically cause HIF to increase in the old rats, it will. But the normal stimulus of hypoxia doesn't produce the HIF. That means the older rat is vulnerable. Obina showed, was an MSTP student, graduated a couple of years ago from Case, showed those black bars decreasing with age of the rat from 3-month-old, 6, 12, 18, 24 months old, a decreasing response to HIF with age. And Guriso showed in mice the same thing, and that half the mice almost died during the hypoxic exposure, whereas normally they all survive. Now, if you do increase, that's bad news, but the good news is if you can increase the HIF in these rats, then they can deal with increased cognitive performance. So the diet produced an increase in cognitive performance in these rats. So again, the neurovascular unit is the key to what's happening with aging, and how to interact with disease. These are the stats from the CDC on stroke. The bottom line costs are about $34 billion per year. The amount of money spent on stroke research is a tiny fraction of this on an annual basis. There is what's called the MOVE AD, which NIH, NIA, and NINDS is trying to do research on capillaries, on vascular control in dementia. Alzheimer's stats, these are through the roof. Bottom line, again, costs are in the $200 billion range. We have slightly increased our funding for Alzheimer's research. That's a good sign, but again, a small fraction of what it costs annually. And as far as a track record goes, we've done, this is the percent change in death rates. Alzheimer's has increased by 38%. We've done pretty good on some of the other death rates decreasing in cancer, heart disease, and stroke, but Alzheimer's disease is still a major, major factor. These are the obvious conclusions of support angioplasticity and support treatment strategies against these diseases. Bottom line, take-home message, capillaries are the link between aging, stroke, and other disease states. Thank you very much for your attention.
You've been watching Joe LaManna talking about the connection between diet and brain health. Up next is Dr. Lynn Singer discussing the impact of environmental toxins on children's health. Now, back to the talk.
The new childhood illnesses, addiction, autism, and lead poisoning. Patricia gave me this title, and it does really come together, bring together a number of toxins that we need to be thinking about in terms of child development. So how can developmental neurotoxicology, I'm a developmental neurotoxicologist in one of my identities, how can that prevent lifelong disorders? And what is developmental neurotoxicology? First of all, study of ology, poisons, toxins, and those that affect the development of the brain, neurodevelopment. And it strikes me today that we're talking a lot about brain illnesses on multiple levels. So, how can we protect millions of children from chemicals that harm their developing brains? What chemicals are we thinking about? And how do addiction, autism, and lead poisoning relate? So we previously believed that the mammalian embryo was protected from all external factors by the placenta. After thalidomide, it became accepted knowledge that the developing embryo can be highly vulnerable to agents that may have negligible or non-toxic effects in adults. And this is very interesting because these toxins have existed for centuries, millennia, but we really didn't put it together. When we believed that nothing could cross the placenta, scientists didn't see that there could be damage due to it. So also for many years, scientists thought about birth defects only in terms of abnormal physical structures, as with thalidomide. However, intellectual and emotional development cannot be determined until long after birth. And we know that brain development, and not just limb or physical development, can be affected. Thalidomide really was for us the big reveal. It was a prescription drug sold from 1957 to 61 in 50 countries to combat morning sickness in pregnant women. 10,000 children were born worldwide with phocomelia, primarily in Europe and Canada. We were lucky. Francis Kelsey in the United States, who was head of the FDA at the time, would not allow it in. There was a lot of controversy at the time. One of the important things to think about of toxins like this, or any kind of chemical that the fetus is exposed to, is that the limits of danger are amazingly narrow. For example, the damage from thalidomide is done only with could be done with only one tablet taken, but only between the 20th and 36th day after conception. If you took it in the third trimester, you were fine. If you took it before that, you were fine. But if you took it during those days, that's when the damage occurred. And it's been very significant in terms of understanding the development of disorders and the progression of fetal development from embryogenesis to physical development to brain organization. Interestingly, over 2.5 million tablets, despite the fact that it was not to come into the country, were given out in the United States despite no approval from the FDA. It was a prescribed drug in Europe and Canada, and many people got it like we can get drugs today. Thalidomide, however, if you think about it retrospectively, the limb defects were the phocomelia was really what we consider. But it was one of the major causes of autism, and that's been not as well recognized. So it's one of the known causes of autism, as is another epidemic that we had, which is rubella and measles. And which I find particularly ironic, and that many people now are fighting against the use of the measles vaccine, when actually rubella, measles, is a known cause of autism. Another historical event that really had a big impact on the scientific community was that in the 1950s, fishermen and their families in the small fishing village of LaManna began to suffer a disabling neurologic disease that eventually was confirmed as methylmercury poisoning. Prenatal exposure seemed to inflict far greater damage than exposure during adulthood. The children of mothers who had eaten a steady diet of bay fish during pregnancy exhibited mental retardation, cerebral palsy, and blindness, even when mothers showed no symptoms. And of course, mercury is a ubiquitous contaminant, but this was one of the first population recognitions of the damage that this exposure could cause. These early environmental tragedies help scientists understand a few key issues. One, toxins can cross the placenta during pregnancy, affecting embryo and cell development and brain organization. Two, toxins can be drugs, prescribed or illegal, alcohol, household products, environmental pollutants, and we'll talk about those. Toxic exposures during pregnancy can result in physical defects, but also in long-term developmental disabilities, such as autism, attention deficit disorder, low IQ, learning or speech problems, and motor delays. So we no longer have a risk of thalidomide in this country or in the world, but our environmental exposures to things like mercury does continue, which is why you're told not to eat a lot of certain fish, especially if you're pregnant. Another example of a major toxic exposure that you may be aware of is lead. National surveys indicate that more than 3 million children 6 years of age and younger have lead poisoning. This slide shows you the levels of lead found in various water samples. For example, Troy, Michigan at 1.1 parts per billion, Detroit, Michigan, double, but under the level of concern, which is 5 parts per billion. Flint water, as you know, was found to be 5 times the safe level. However, if you look, the highest level was actually 1,000-fold higher than safe. So lead exposure, we know, has multiple effects on the body, but particular effects on the developing brain, and has been linked to low IQ, attention deficit disorder, hearing loss, and behavioral problems. We're also demonstrating over time, over the last 20 years, a number of scientists have shown that it used to be that the level of 10 was considered to be a safe level. We now know that there are effects even at the level of 5, and you can continue all through that continuum of looking at that. Interestingly, you'll hear my studies have been on cocaine exposure in Cleveland, and even after we screened for lead, in our studies we found that 25% of children born in the 1990s in Cleveland, in the city of Cleveland, inner city of Cleveland, had high lead level exposures over the 10, over the risk limit. So we know that these are now effects on the developing brain. They can occur prenatally if the mother is exposed to lead and has lead in her body, as well as postnatally. By the 1970s, scientists began to understand that these toxic exposures not only led to physical changes, but to behavioral and functional disabilities, as we said, due to their impact on brain development. Here we see the first known and studied patient with fetal alcohol syndrome, characteristic poor growth, unusual facial features, and mental retardation. But FAS, fetal alcohol syndrome, is only the tip of the iceberg. We now know that even without the physical defects seen in frank fetal alcohol syndrome, there are effects at lower doses. We have variously identified them as fetal alcohol effects or alcohol-related neurodevelopmental disorders. Recent studies, for example, have demonstrated that subtle cognitive and attentional deficits can be found in non-fetal alcohol syndrome children born to moderate drinking mothers. And the field is now searching. We do not have a known safe level of alcohol use during pregnancy. And I think back to Michael's thing. We often think, so many people drink and everything is fine, just like some people smoke cigarettes and don't get lung cancer, and also some pilots can endure the hypoxia and not have the effects. But there are known across the population effects, and this is one of the studies that demonstrates it. For example, you can actually see an actual dose-response relationship here with the children of mothers on the left who never drank having IQ points 8 points higher than those of mothers who continued to drink during pregnancy. And these were not alcoholics, but normal, moderate drinkers. We also know these are some of the disabilities that go along, and they go along with all of the brain disorders, the level, what are involved in developmental disabilities, which are rising in our children's population today. And autism is kind of the poster child for that. But the disabilities that are affected by these toxins, alcohol, and particularly in alcohol, have been identified: cognition, memory, attention, perseveration, and the modulation of the sensory systems, and planning, and what we call executive function. So, what other kinds of toxins are out there that are affecting? Millions of young women of childbearing age consume illegal drugs, and many become pregnant. Some continue to use during pregnancy, especially if they're dependent, and many studies now demonstrate the effects of these drugs on offspring outcome, primarily on the kinds of things you saw with fetal alcohol, motor, language and learning problems, motor coordination, executive functioning, and attention. And so even though there are multiple chemicals, there seem to be common pathways, and it all depends on what stage, the amount, the dosage that the infant was exposed to or the fetus was exposed to. These are all brain illnesses. My own studies have focused on cocaine and MDMA or ecstasy. There are a number of longitudinal studies going on now looking at methamphetamine. There's going to be a large study on marijuana. We need many, many more of these studies. Of course, these are illegal drugs, and they're used by millions and millions of young people. But we can also think about things like some of my studies involve antidepressants. We do have concerns about selective serotonin reuptake inhibitors and what happens to child development after exposure to those drugs. And they also have been linked in some studies to autism. Opioids, as you know, are another drug that seem to be many, many women have become dependent on, and we really don't have any data, very, very little data on how those drugs affect child development. Exposure to toxic chemicals during pregnancy is ubiquitous. The National Cancer Institute report has found that, quote, to a disturbing extent, babies are born pre-polluted. Exposure to air pollution and to pesticides have also been related to a higher risk for autism and developmental problems in scientific studies. Another environmental exposure that has been investigated prospectively is polychlorinated biphenyls. These are PCBs. They're now banned, but the residue, the sequelae, are ubiquitous in our environment. They used to be used in electrical transformers. PTBs and flame retardants are another one. These also have been banned, but what happens is the chemical industry takes a variant of it, which is not deemed unsafe, but we don't know, but it's very highly related, and we can't really tell. Until we prove that it's unsafe, we can't get it off the market. So the top 6 toxic threats worldwide, as you can see, are lead, mercury, with some of which we've talked about, chromium, arsenic, pesticides, and radionuclides. And these are affecting millions of pregnant women and offspring in some ways that we know about and others that we don't. We talk about autism. Autism is a complex disorder of brain development, which is now affecting by the CDC standards, 1 in 68 American children. It's a broad heterogeneous category of developmental disability. But as you can see, all of the kinds of things that we've talked about, intellectual disability, difficulties in motor coordination and attention, these are all the kinds of brain functions that have been affected by many different kinds of chemicals. What are the causes of autism? As we said, there's genetics, there's epigenetics, but then it's also how the environmental risk factors affect those genes structures. We know genes don't change as quickly over time as the environment seems to have changed. And because of the rapid rise in these developmental disabilities, such as autism, there's a question, and there have been links to a number of our environmental chemicals that I've been identifying for you that can affect the fetus before and during and after pregnancy that have been rising. So we think that that has a possible impact. So the bottom line, many chemicals can interfere with healthy brain development prenatally and during early developmental stages. These are toxins. They can be alcohol, illegal or prescribed drugs, or environmental chemicals such as lead, pesticides, PCBs, mercury, or air pollutants. All have been related in various ways to the increase in developmental disabilities that's occurring. Low IQ, autism, ADHD, learning disabilities. These have staggering economic costs in the billions a year to our economy. Learning and behavioral disorders. These are learning and behavioral disorders that can be prevented or lowered by reducing or eliminating toxic exposures. We know that fetal alcohol syndrome is 100% preventable, and the mental retardation that goes along with it. It's the leading cause of mental retardation. And finally, there is an urgent need for using scientific evidence to change our public policies to reduce these toxic exposures. There's a group of scientists who've come together recently, you may have heard about it in the newspaper, called Project Tender, which has been focused on these air pollutants and chemical exposures, many of which I've named, as major potential linked causes to developmental disabilities in this country, and calling for really scientific understanding of these disorders and a call to action for all of us to prevent these disorders. So thank you.
I was cleaning out our medicine cabinet the other day. We had a whole bunch of medicines that were 5, 10 years out of date. And I didn't know what to do with them. Actually, I tossed some in the garbage. I shouldn't have done that. But what are you supposed to do with old chemicals like that?
What I understand that you're supposed to do is kind of wrap them up in a container that can, there are places where you may take them possibly in the city of Cleveland, but if you can't do that, you wrap them up into like a plastic container so that they can't come out, and you put them in the trash. You can go on the internet and see exactly the recommended method for disposing of them. Do not put them down your toilet, because then we all drink the antidepressants or whatever they might be.
So is this an issue only after you're pregnant, or is it also both for the father and the mother exposure before?
So actually it's very interesting, because we know that certain things, for example with drugs of abuse, like cocaine or like MDMA, in one of the studies I did, we actually found that the report of use of those drugs earlier, because what happens is that when you use a drug of abuse, it affects the neurotransmitter systems in your brain. So for example, MDMA affects the serotonin system. And that means that when someone takes MDMA, 80% of their serotonin is depleted and may do downregulation to that system, which is really very important for fetal development. It's the first neurotransmitter in existence, and it kind of sets off the whole cascade of development. So that would mean a mother is going into that with a probably very depleted serotonin system of her own, which will affect the development. The other thing I did not mention, and that's the other half of this birth equation, is the father. And there's increasing evidence that fathers' use of drugs, et cetera, also, we certainly have seen it with alcohol, There's an effect on child development based on the father's use of alcohol. And even though it's not during the pregnancy, it does affect sperm, in some way. So that but that's totally almost non-studied subject. But it makes sense. Think about it. That genetic pool over there is certainly affected by various kinds of drugs as well.
You've been watching Lynn Singer talking about the links between the fetal environment and childhood illnesses. Up next is Dr. Scott Simpson discussing recent advances in our understanding of early human evolution. Now, back to the talk.
So I'd like to talk a little bit about human evolution, and that the topic is, recent advances. And what is going on in human evolution that within the past 30 years is that there's been a tremendous amount of change going on in terms of fossil finds and using different approaches. And what I'd like to do is just give you a thumbnail sketch of some of the changes that have been going on in the field that have really improved our understanding of the past and really expanded our understanding of the biology and the context of human evolution. Do we have... here we go. So in talking about these recent advances, some of the things we can talk about is that we have some of the earliest human ancestors. When I started graduate school about 30 years ago, human evolution stopped about 3.5 million years ago, in the sense that we didn't have any understanding of the ancestors that went back older than 3.5 million years ago. Now what we do is we have fossils that go back to 6 and 7 million years ago, which we think are human ancestors, and plus we now have genetic data that allow us to go back and reconstruct the timing and patterns and geometry of speciation between humans and our earliest, or our nearest, closest living relative, chimpanzees. Other things that are very important is we can understand the origins of bipedalism, walking on two legs. Now this might seem that, why do we explain this? This needs explanation because we're the only mammals that can walk on two legs. And as we'll see when we talk about bipedalism, even though it means two-legged walking, is in fact the major adaptations are to being able to stand on one leg. Another thing we can look at is increased species diversity. What's in our own personal experience, there's only one flavor of Homo sapiens. It's us. We have regional variations but we're distributed all across the world, and there's just one species of human. And so it's many times very difficult to understand that in fact there were different ways to be humans or human ancestors. And the fossil record is really illuminating how many differences have occurred. Another thing is evolutionary medicine. Now, what we can do is, because we have this historical perspective, we have fossils that go back 5 or 6 million years, what we can do is we can look at the patterns of change in our anatomy and biology over that time period, and we can use that as a reference to figure out why humans have certain health problems that we have today. And so it's a very useful adjunct to understanding patterns of disease. And then I'm a paleontologist, and so I'm used to thinking about things in terms of fossils, bones that you can pick up, teeth that you can pick up. But what's happened now is that with the ability of researchers to isolate and amplify ancient DNA, in fact, now we're starting to use DNA in our paleontological studies. So for today's talk, what I'd like to do is talk about some recent advances in the origins of bipedalism, the fossil record for that. We'd also like to talk about a little bit about evolutionary medicine in terms of childbirth, and also recent advances in ancient DNA. So of this is the best way, because paleontology is an historical science. And I'm always hoping that the best way in which we can go back into the past is just develop a time machine. Now, unfortunately, time machines don't exist. But if they did, I would tell Sherman, set the Wayback Machine to 4.4 million years ago so that we could begin looking at some of our earliest ancestors and see how they moved. Because that would be the best way to understand our early biology. We could observe our ancestors, we could see what they ate, we could see where they lived on the landscape, when they died, we could dissect them so we could understand the nuts and bolts of their anatomy. But we really can't do that with, in the absence of a time machine. But what we do have is a time machine. And this time machine is fossils. And fossils, as you know, are direct evidence of the individuals who lived in the past. These are their bones and teeth or their footprints, any evidence that we have. And we have to understand that without paleontology, without this fossil evidence, we would never know anything about trilobites. We would never know anything about Tyrannosaurus rex. We would never know anything about dinosaurs or any of the other primitive mammals or any of the ancestor forms that are no longer living today. And so that's why these fossils are so very important, because they allow us a means of looking into the past. Now, originally I made this slide 10 years ago, and really focused on, emphasized the paleontological aspect, but really have to add the DNA aspect to it because there are great advances in being able to isolate DNA. And it's and the story that the DNA is produced is much more complex and much more interesting than anybody had anticipated. So I'd like to talk a little bit about why this is a golden age of paleontology, human studies. I'm going to focus on Africa, but there's a tremendous amount of research that's going on throughout the world, but just my experiences in Africa. And so what we have here is different localities and what we knew in terms of places we were finding very ancient human ancestors. And before about 1959, 1960, we only had a handful of sites where we were recovering these fossils, and these were from South Africa, and they weren't very well preserved. But in the intervening years, what we're doing is we're finding more and more and more fossils throughout Africa, although I'm, again, focusing on the Eastern African rift system, but there are also fossils that are being recovered in Central Africa, Northern Africa, and Southwest Africa. And so we can see that as we're finding all of these additional sites, we're learning so much more about the population of ancestors that lived over these past 6 million years. So again, going back to 1960, this was the spectrum of human variation that we had observed. We had 5 or so species of named ancestors. And on the left column was a vertical column is about this time. And we didn't know back in 1960 or 1965 how old these fossils were, so they could be linked in any sort of way. And so what we did is we thought we could link all of these fossils into one single lineage, going from the green Paranthropus robustus on the right to which we thought at the time was the more derived Australopithecus africanus, up to Homo erectus, Neanderthals, and then modern humans. But as we saw with the increased field research going throughout Africa and across the world, we're now starting to identify lots and lots and lots and lots of different species, as you would expect. And as you can see, we're now also starting to find human ancestors that are going back over 6 and 7 million years ago. These are very primitive organisms, or ancestors, and very interesting anatomy. So in today's talk, what I'd like to do is focus on 3 of these species that have been new interpretations or newly described species. This includes the 4.4 million year old Ardipithecus ramidus. This is a fossil species, a human ancestor that was initially discovered in the 1990s. It's been found in 2 or 3 different localities in Eastern Africa. Then we'd like to go back and have another look at one of our old ancestors, Homo erectus. Everybody's heard Homo erectus, Peking man and Java man, etc. And we'd like to have a look at how that in biology informs our understanding of our biology. And then we'd also like to look at the Denisovans. And the Denisovans are a newly identified genetic species. So beginning with this, we're talking a little bit about the recent discoveries from Ethiopia, where I'm most familiar. I've been working in Ethiopia since 1992. And this satellite image on the right is from the Afar Triangle. It's in the Afar Depression. It's one of the hottest places on Earth. The area just off the north of the image is called the Donakill Depression, which National Geographic calls the hottest place on Earth. But even though it's very hot and arid today, what we find is in the past, over the past 6 million years, this was a very, very rich area of rivers and lakes and tremendous diversity of plants and animals that were living there at the time. Importantly, what we find is that there were a number of human ancestors that were also living in the time. And why that is important for us today is because not only were they living there at the time, it's through the geology that's been occurring as Africa is being torn apart, these fossils are now being re-exposed so that we can go out and collect them and find them again. So there are each of those names up there is a different project area that's currently working in Ethiopia. But the 3 we're going to focus on here are Hadar. Perhaps many people have heard of Hadar. That's the home of Lucy. Gona, which I've been working since 1999, and then the Middle Awash, another place where I worked in throughout the 90s. So the first one I want to talk about is our unexpected ancestor. And Ardipithecus ramidus is the name of this ancestor and the origins of bipedality. And so in order to better understand the origins of bipedality, we have to refer to something everybody's familiar with, and this is the Lucy skeleton. Lucy is very popular here in Cleveland, as you know, because one of the members who led the team in the discovery and analysis of Lucy was based here at Case and at the Cleveland Museum of Natural History, and the core analytic team was based here in Northeast Ohio. So Lucy was very important because she was a reasonably complete skeleton, she was very well dated, and she's beautifully preserved. And so as a consequence of this beautiful preservation, what we can do is we can then reconstruct this pile of bones into a coherent skeleton, and then using our understanding of soft tissue anatomy, we can then build this up to a single individual. Now, the important aspect of Lucy was the fact that she was so beautifully preserved, but she preserved evidence in her musculoskeletal system that she walked bipedally on two legs in the same way that you and I did. Her fingers were a little longer than ours, her legs were a little shorter, but in terms of how she moved around, she walked just like we did. And so if we're not sure, let's say we blew the anatomy of this and we completely misinterpreted this, in Laetoli in Tanzania in the 1970s were recovered a series of footprints preserved in a volcanic tuff, which clearly show three individuals walking together across this plain on two legs. So this brings us to Ardipithecus ramidus, because the idea is what was the antecedents of this bipedal walking? Were we bipedal forever? Or what did an early biped look like? And beginning in 1994, this skeleton, partial skeleton, which we nicknamed Ardi, was discovered in the Middle Awash Project area by Johannes Haile Selassie, who was a curator of physical anthropology at the Natural History Museum. And this, again, is a reasonably well-preserved skeleton. And we were, through our understanding of anatomy, could build up an analysis of the skeleton. And we can see that she's very different from Lucy. And the fact that what she did is, if you look at her foot, is the most interesting aspect, because her foot, her big toe, instead of being in line like ours is, it's off to the side. So it was a grasping organ as in chimpanzees and gorillas, and not the propulsive organ in the same way it is in humans. We can also look at this in terms of her anatomy of her pelvis, which is central to understanding of locomotion. So if we look at the top of the ilia portion, which is that bump that we feel on the side by our belt, we can see that in humans it's twisted and lowered relative to the chimpanzee on the right. Ardipithecus ramidus at 4.5 million years ago also had this anatomy. But unlike modern humans, Ardipithecus also had a very chimpanzee-like lower pelvis. And consequently, we can break this down so that the upper pelvis, which we refer to as the false pelvis, or the gluteal portion of the pelvis, is very human-like, meaning that Ardipithecus could stand on one leg like we could. But the lower part was more like a chimpanzee or other quadrupedal primates, indicating it was still climbing adaptation. So what we've done is we found this intermediate form that shares a mosaic of adaptations that's much older than Lucy. So we now can get back to what the earliest levels and contexts were of the evolution of our bipedality. So switching gears, we'd like to talk then a little bit about evolutionary medicine. And this is, again, understand how our history, our biological history, can inform patterns of disease in moderns. And so the bipedality was a major adaptation. It changed our entire musculoskeletal system from the tips of our toes to our base of our cranium. We are completely reorganized. And as a consequence of changing our ancient quadrupedal skeleton into a bipedal skeleton, it resulted in a number of changes that then affect us now, including we have bad backs, we have bad knees, we have foot problems, we have things like hemorrhoids, we have hernia problems that other animals don't have. Now, if you want to look, but the one thing we're going to talk about here is the increased difficulty in childbirth. Now, childbirth, you'd figure, is central to our adaptation. Remember, about natural selection, the success of being a successful individual is producing as many individuals as you could possibly produce in your lifetime that survive to adulthood. So why do we have problems making babies? Locomotion. Because as we became a biped, as we see here on the left, what this did was this caused a whole complete change in our musculoskeletal system, but especially in our pelvis. And so what makes a good bipedal pelvis or a monopodal pelvis versus a quadrupedal pelvis makes it a bad obstetric or baby producing pelvis. So the opening that we see that's in the central of the pelvis, that's the bony birth canal. Chimpanzees have a large one, humans have a small one. And we can emphasize how small this is as a consequence of when we try to get a baby through, babies get stuck because they have big heads, they also get stuck because of their shoulders. So a consequence of this is humans have difficulty in childbirth for two reasons. One, we had to reorganize our pelvis in order to become a biped, and second, we've got big brains. And so it's the big brains are also a real significant contributing factor to why babies get stuck in the birth canal. And if we just look at a quick evolution here, so chimpanzees on the left, humans on the right, this is little Lucy's pelvis at 3.2 million years ago. And Lucy had a locomotor pelvis just like ours. All the musculature was oriented in the same way. But if you look at the hole in the middle, it was very small. Now for Lucy, it didn't matter too much because her offspring were quite small. However, as our brains became bigger, especially between 2 and 1 million years ago, what we had to do is we had to increase the size of our bony birth canal. And so this is a pelvis from Gona in Ethiopia, and we can see that the birth canal has increased in size, not quite as large as modern humans, but we can see that there's this coevolution going on between an increase in the pelvis and an increase in birth size, which again in some circles provided a revelation. And then finally, what we'd like to do is talk about what I refer to as our invisible ancestors and DNA in the Denisovans. Now, if we look at the paleontological record and the archaeological record across Eurasia, especially in Western Europe, what we find is that between about 500,000 years ago or or longer than that, till about 40,000 years ago, what we find is just one flavor of human ancestor, and this is Neanderthals. Now, beginning about 40 or 50,000 years ago, moving into Europe is our ancestor, us, Homo sapiens, which arose in Africa probably about 200,000 years ago and left Africa about 60,000 years ago. And an important datum for this evolution or this migration of our species out of Africa is the Manat Cave in Israel, which provides evidence of some of the earliest Homo sapiens in that region. Very interesting anatomy on that specimen as well. But what we also find is that, so we have good fossil evidence for these two individuals, Neanderthals and Homo sapiens, but then we also have something else, Homo spa, we don't know, which we nicknamed the Denisovans. And we don't have a picture yet because there is no picture available. So let's look at how DNA has affected, our understanding of DNA has affected how we work in paleontology. First off, the dark gray area is the known distribution of Neanderthals, where we find their bones and their tools. The gray area off to the right in a site called Akladnikov is a site where they found these, small fragments of humerus and femoral bones, but were unidentifiable, but they had DNA. They analyzed the DNA, and what they did was they found that it was just like the DNA, the mitochondrial DNA that was discovered from the Neanderthals. So what this did is, even though we didn't have a good characteristic or diagnostic Neanderthal fossil, we had the diagnostic DNA, and it expanded the range of Neanderthals 1,500 miles further to the east. Right next to the cave, or nearby the cave of Akladnikov, is another cave called Denisova. Denisova has also produced some fossils. And Denisova is dated to about 50,000 years ago, right in the later part of Neanderthals, and it produced a small toe bone. What's interesting about this toe bone, we could get, because it's a cold, dry cave, we could get DNA from this, and the DNA from the Denisovan showed, okay, it's just like a Neanderthal, so this is what a Neanderthal toe bone looks like. But immediately below, in a layer also dated to 50,000 years ago, are a couple of other small bones, a tooth on the bottom and a small fragment of a finger bone. When the mitochondrial DNA of these specimens were analyzed, a very different story arose. So let's look at the cave. So we have the toe bone up there in blue, compared the mitochondrial sequences, mitochondrial DNA sequences with Neanderthals, and you can see that they all group together very closely. And then when we compare this with humans, we can see that Neanderthals are a coherent group that is different from that we see in the modern Homo sapiens. But then when we analyze the tooth and the finger bone, we get a very different DNA pattern. It's very, very different from the Neanderthals, and it's very, very different from Homo sapiens, suggesting that we have identified, not based on anatomy of the morphology, but we've identified based on the DNA, a new species, which we now call Denisovans. No one has yet to name it, assign it a true species name. And so just in comparison, when we take other DNA evidence into account, we can look at, in fact, that there are 3 flavors of humans that were living in the Eurasia area. After about 60,000 years ago, we get modern humans, but before that we had 2 species, the Denisovans, which are represented by 2 fossils, or well, just a very small handful of fossils, and the Neanderthals. And I'd like to say that we could continue this story because it's absolutely much more complex and much more interesting than I've presented here. But we're going to find that the DNA analyses are going to be essential to really unscrewing the unscrutable about the very complex evolution of humans across Eurasia. And so what I'd like to do is, as a paleontologist, we always think about the beginning with the bones, but we want to make sure that we have to translate this into something that is real, in our case, our ancestors. Thank you very much.
The Real Science Now lectures are hosted by the Great Lakes Science Center and presented by the Institute for the Science of Origins with the assistance of Case Western Reserve University, its College of Arts and Sciences, and MediaVision. For more information on Real Science Now and the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.